Hello everyone, I'm back with our new topic, which is on movement in and out of cells, chapter 3. So in this chapter it's going to be exercises based mainly on cells and experimental and graphical questions actually. Also on diffusion, mainly on diffusion. There's like a whole exercise dedicated on diffusion actually. So let's begin off with the definitions. So the first definition, as you can see, is diffusion. So it's the net movement of molecules and ions from a region of high concentration to a region of low concentration down a concentration gradient as a result of their random movement. And then for osmosis, it's supposed to be the diffusion of water molecules from a region of higher water potential to a region of low water potential through a partially permeable membrane. So guys, don't be confused that for diffusion process the molecules and ions need partially permeable membrane. This only applies to osmosis. It's very easy to confuse between diffusion and osmosis, as it's almost relatively the same process from a higher concentration to a lower concentration. Whereas for active transports, it's literally the opposite. It's like the movement of molecules and ions in and out of a cell through the cell membrane against a concentration gradient using energy from respiration. Meaning for active transport, it goes from a region of lower concentration to a region of higher concentration against a concentration gradient, and this process uses energy from respiration, which is a chemical process that occurs in cells. Whereas for diffusion osmosis, they don't use energy at all, they don't require energy at all. Happens from a higher concentration to a lower concentration. Whereas for active transport, it happens from lower concentration to high concentration. Right, so let's move on to the main point, the main exercise 3.1, diffusion experiment. This exercise asks you to handle and interpret data collected during an experiment, also to think about how the experiment was planned. A student did an experiment to test this hypothesis. The higher the temperature, the faster the diffusion takes place. So let's see what the question is about. She took 4 petri dishes containing agar jelly. She cut 4 holes in the jelly in each dish. She placed 0.5 cm cubic of a solution containing a red pigment into each hole. So this is the agar jelly, and this is the holes, 4 holes, okay, that are being cut in the jelly, and this jelly contains a red pigmented solution. Right, so the student then covered the dishes very carefully and placed them in different temperatures. She left them for 2 hours. Then she measured how far the red color had diffused into the agar jelly or into the agar around each hole. The table shows the students' results. Dish A, dish C, and dish D. So there are about 4 dishes at different temperatures, because they want to know how far the pigment traveled on at different temperatures actually. So distance, red color had diffused in the jelly, hole 1, hole 2, hole 3, hole 4. So all these 4 holes, they were all kept under different temperatures. So for hole 1 at temperature 10, the distance traveled by the red color was 2 mm, hole 2 3 mm, 2 mm, 3 mm, 10 degrees Celsius, which is quite very less. 20 degrees Celsius, 5 mm, 5 mm, 6 mm, 4 mm. 40 degrees Celsius, 9 mm, 11 mm, 8 mm, 10 mm. 80 degrees Celsius, 19 mm, 21, 18, 23. So as you can see, the higher the temperature, the faster the diffusion takes place. So this hypothesis is actually correct. Even in reality, in real life, it's actually correct. If you guys can test out, it's the same thing. For example, if you have like a milk, or you could say if you have like a milk and you have a chocolate powder, and if you warm the milk, okay, and then you mix the chocolate powder inside, the chocolate powder will diffuse way more faster than in cold milk. So the same analogy applies over here as well. So now, part A. Complete the table by calculating the mean distances diffused by the red color in each dish. Give each distance the nearest whole number, because this is how the student measurements were taken. Write your answers in the table. Okay, so now they give you one tip over here. When there's any question asking you to give the average or give the mean, okay, your answer should be in the same format as it's given in the data of the question. So since all the data is in whole numbers, so your answer should be in whole number. If it's one decimal place, your answer should also be in one decimal place. In the real IGCSE question paper, they will never give you such hint, so you guys need to take note of this. But sometimes they actually give an exception, even if the data is in whole numbers, but your answer is like one decimal place, they still have a compensation on that as well. They're not that strict, but still it's fairly okay. So it's quite easy to calculate the mean. You have to like first add up all these, then divide by 4, add up all these, divide by 4, add up all these, divide by 4. So make sure they're all in whole numbers. So this is 3, 5, 10, 20. Now part B. Do the results support the student's hypothesis? Of course it does. Why? So if you see over here, as the temperature increased, the distance the red color diffused through the jelly also increased. So as the temperature increased, the distance diffused also increased. As the dishes were all left for the same period of time, they were all left for 2 hours, okay, so this means the color was moving faster in the warmer dishes. So as you can see, at 80 degrees Celsius it was 90 millimeters, with 10 degrees only 2 millimeters. A doubling of the temperature caused the distance diffused by the color to roughly double. This is actually not, it's one word. A doubling of the temperature caused, so you see it's like times 2, times 2, times 2, okay, doubling of the temp caused the distance diffused by color to roughly double. So basically, as you double the temperature, the distance diffused will also double, and as the temperature increases, the distance diffused also increases. And since it's actually a fair experiment, it was left at 2 hours, so I wouldn't say it's fully fair experiment, but since they kept it at 2 hours, the same duration of time but different temperatures, meaning the distance it diffused were actually faster in the warmer temperature than the colder ones, right? This is roughly around, like, say 2 marks. They want you to write down to prove that as the temperature increases, the distance diffused is also increased, and doubling the temperature causes the distance diffused to be doubled. So now, state 4 variables that the student kept constant in her experiment, or that she should have kept constant. So the first thing you should know, in the whole question, they didn't say the concentration used of the pigment. They did not mean, they just said that she placed 0.5 centimeter cubic of a solution containing red pigment into each hole. So they actually mentioned about 0.5 centimeter cubic of a solution containing red pigment into each hole, meaning each hole had like 0.5 centimeter cubic, right? So actually in the question, like, the only thing that they kept constant is the duration of time. And since the question is basically asking you to find out what were the variables this student kept constant, so you can mention this one, she kept the concentration of the pigment, the duration of the time, or that she should have kept constant the size of the hole. Yeah, it's just holes. She just cut 4 holes in the jelly. We don't know which hole was bigger than the other hole, which hole was smaller. So the one that she should have actually kept constant was the size of the hole in the jelly, depth of the jelly in the dish, since they were like 4 dishes. So we don't know how deep the petri dishes or how deep the agar jellies were. So she should have like maintained the depth, and the volume of solution placed in the hole. This is just the concentration. 0.5 centimeter cubic is the concentration. Like how concentrated the pigment is. there is no they didn't state how much volume of the red pigment was put into each hole. We just know how concentrated the red pigment was. And another thing you can mention about the duration of the time was same 2 hours, right? So these are like 4 things. So another 5th thing you can mention is about you could say duration of the experiment, 2 hours. Since the question is asking you the variables that the student kept constant, that means you can use the same thing present in the experiment over here in answer. But if the question asks you, say 4 variables that the student should have kept constant, meaning those variables that she did not, those variables that were not mentioned in the question, you have to mention it. But since they say or, so you can do either ways. So explain why it was a good idea to have 4 holes in each dish rather than just one. Of course, the first thing that should come in your mind, why there's more more things than just one, is because to improve the reliability, to improve the accuracy. So in this way you can calculate the mean, or you can say it improves the reliability. So this is like one key term, reliability, or you can say it allows the mean to be calculated. Now, suggest 2 significant sources of experimental error in this investigation. So now if you find error, since it just says suggest, you don't have to explain, but I actually mentioned it over here why you have to state this. So measurement of the distance diffused. Why? Because the edge between the color and the uncolored jelly will not be very distinct. So one thing you can mention is measurement of the distance diffused. Right, so you could just say the measurement of the distance diffused, and then the time taken for the dye and gel in each dish to reach their final temperature. Time taken for the dye and the jelly in each dish to reach the final temperature. The dye won't have been at the correct temperature for the entire duration of the experiment. So basically what they're trying to say is that the dye, which is the red pigment, okay, the starting temperature could have been different in each holes actually, right? So basically these are like 2 things you have to mention. Measurement of the distance, like basically asking experimental error. So measurement of the distance diffused, although they mentioned over here how long the red pigment diffused, but you know, the edge between the hole and the like in this hole it contained the red solution, but you know, sometimes when you put the red solution, it could be little bit around the outside the edge of the hole. One hole, like it could be like properly all the solution, the red pigment could actually be fully into the hole. Some holes the red pigment may be outside slightly around the edge, so it kind of not that accurate actually, right? So you, like, you know, initially when you cut the hole and when you add in the red pigment, it may slightly be outside the hole, outside the edge, so that causes the error in the experiment. And time taken for the dye and the jelly in each dish to reach the final temperature. Yeah. So now exercise 3.2, how plants take up water. So this exercise checks that you haven't forgotten about cell structure. This is so important to know the cell structure in both plants and animals, the main differences between plant cell and animal cell. You have to know this because it's a very popular question. They almost ask every year. So it also develops your ability to use knowledge in new situations. So now plants take up water into their roots from the soil. They have tiny hairs on their roots which help them with this. The hairs are called root hairs, and each one is a part of a single cell. The diagram shows root hair cell. Okay, this you have not learned about the root hair cell because that's in chapter 6, which is called plant nutrition. you will learn that later on. I don't understand why they include this root hair cell in this chapter, because when you look at the diagram you like just freak out, like what the hell is this? I know it's completely normal to freak out for very new diagrams, because you guys haven't learned this diagram for this chapter 3 yet. It will be in chapter 6. But basically a root hair cell is basically a cell that is present in root hairs, like you know a plant that has roots. Basically that root, there are obviously cells present in the root, so you call it root hair cell. And in that root hair cell, the function of root hair cell is to absorb the water from the soil and other essential nutrients. So that's the function of the root hair cell. So a root hair cell is very similar to a plant cell. Like the only thing that's not present in root hair cell is the chloroplast. Right. So now state 2 structural features of this cell that are typical of plant cells but not animal cells. So basically they want you, what's the in simple words, like they want you to state what's similar of this cell, like what's the similarity between this root hair cell to a plant cell. So in a normal plant cell you have a vacuole and you have a cell wall, but animal cells these two things are not present. However, in a plant cell you have chloroplast as well, but over here there's no chloroplast, so it's not a similarity. So they're typical of plant cells, meaning they want you to write the similarities between this and the plant cells. So you're at cell wall and large vacuole. So I'll explain the next part of this in the next part of the video, so stay tuned.